Welcome to another edition of Journal Club for the Advanced Science Academy at Morris Catholic. Siena Yannotti will be talking about cannabis and COVID-19 because when you have COVID-19, which I just recently got over, you breathe so well you want to interfere with that by smoking some pot. So, without further ado, get us high, Siena, with knowledge.
Okay, so this paper is obviously about cannabis compounds and how they affect anti-inflammatory activity in vitro, which is like in a lab. It hasn't been tested yet in humans or in any like animals, so it's only lab related. And it's mostly related to lung inflammation, which is obviously a symptom of COVID-19. Okay, so the first slide is regarding interleukin 6 and interleukin 8, which are pro-inflammatory cytokine, which is a type of signaling molecule that is secreted from immune cells, and they are also an anti-inflammatory myokine. The control shows that there is a low level of both interleukin 6 and interleukin 8, and when TNFA is introduced, which is tumor necrosis factor, interleukin 6 and 8 are increased. Also, when dexamethasone is introduced, interleukin 6 is decreased and interleukin 8 is increased. And when cannabis is introduced, fcbd is 66% phytocannabinoids, and fcbd STD is a combination of different phytocannabinoids. So they're all treated with the strain of cannabis that's called C. saveda arbol, and they're all crude.
Hold on a second. I'm sorry. Haley, you cannot be driving with this pointed at you, because if you get in a car crash, then we're going to have issues. I am on the record. Turn off the video and listen to us. There we go. Good Lord. Okay. Yeah. Keep going.
Okay. So that's what's seen in figures A and B. C, B, E, and F are all the dose effective curves of FCBD and FCBD STD.
So I just want to go over something real quick.
Yeah.
So those interleukins are a type of cytokine. Cyto means cell, kine or kine means to move. And so we're talking about in an inflammatory reaction, these signals. So if any of my APs were here, I don't think there are any sophomores here at all, as usual. It's that anxiety just gets them. They can't even come on. We're doing immune system right now, and so in the immune system, in the innate immune system, your first line of defense when something invades your body are these mast cells and macrophages, which produce all of these cytokines. Interleukins are just one of those. So cytokines are also used in the nervous system to give you this sensation of pain. Okay. All right, keep going.
Okay. So moving on to
call on people if you want somebody to do anything. As Lila panics.
So in figure 2, there you go. So in figure 2, it's showing the effects of different dosages of FCBD and just FCBD and CBD, and how higher doses and lower doses affect the amount of interleukin 6 and 8. You can see that they're all like relatively steady, except for what is that? Except for 3 milliliters, the rest are relatively steady. And that the treatments with CBD are significantly higher than dexamethane, but less than TNFA.
So that 3 micrograms per mil should correspond to the previous figure's titration point, right? So fcbd log by, you can't really tell from the concentration there. That should probably be the inflection point, I would think. Right? That sound right?
Yeah, that sounds right.
Okay.
So in table 1, this is just outlining the composition of the phytocannabinoids used in this treatment. So like CBD is 93.5% phytocannabinoids, which is like 61.7% of like the total makeup of it. And the terpenes are produced in the cannabis stevia plant and are part of the treatment of lung inflammation and is a treatment for other medical conditions.
I just want to be clear. Stevia is a type of sweetener.
I thought that was stevia.
It's pronounced both ways. And sativa is the strain of marijuana. But, well, keep going. But I before you move on to the next thing, I want to rail on the medical marijuana community for a moment.
Okay. I don't have anything else for this.
Okay, so let me rail on the medical marijuana community. How many of you have ever been to a medical marijuana dispensary? That better be a no one ever. Hit the raise hand function if you've ever been there legally. I don't want to know if you've.
I've seen like 50 of them. You know, I see these like trucks that you know, like a Weed World or whatever.
Oh no, I mean been in the building which then distributes it.
Yeah.
One of the strangest things I've ever experienced when I was at Brown, we had to do a thing about this, and I walk in and it's like you're in lockdown. You have to go through a what's called a man trap. It's kind of what they designed at the front of Morris Catholic, and you have to be let in one by one, as if you're going to see the wizard or something. And you walk in, and this is medical for medical purposes, and there's Bob Marley on the wall, and all of this like peace, love, hippie crap. And I'm like, if you're trying to sell this as medication, then you got to take that cultural influence out. Similarly, we've done a pretty good job isolating CBDs, cannabinoids, as the active ingredients that seem to work well for inflammatory responses and things. Fine. You could breed out of the sativa or what's the other one called? Indica? Is that the other breed? Indica?
I'm not sure.
Well, I'm sure somebody knows and doesn't want to admit it. I think it's indica. But you could breed out their production of THC, yet they never do. Can't imagine why they don't do that. So, I don't know. I think some of it's a load of crap that it's like trying to provide a medical reason for getting high, which is not medical. So CBDs, cannabinoids, are the things that seem to have the most impact. Okay, go ahead.
So this is showing the treatments of CBD in association with other cells and things found in the body that also fight inflammation. So in figure A, you can see how different like the TRPA1 blocker and FCBD, like in accordance with each other, how they affect the interleukin 6 rate. And there's also just like the rate, how it affects interleukin 6 without CBD as well, in both figure A and B.
Can I point out to the freshmen, look at how they did the A's, B's, and C's, exactly how I talked about it in your lab. And it says, hold on, I'm going to switch to this view so you can see on my version of the paper. But it says here, bars labeled with different letters are significantly different from all combinations of pairs according to a post hoc test, just like we did. So what I told you guys to do is exactly what's used in the literature all the time. Okay, go ahead.
Okay. So, this shows the CCL2 fold expression and the interleukin 7 expression, as well as CCL7 and ACE2. It shows, again, CBD in comparison to dexamethasone and TNF-A, and how it compares to those two, like the steroid that is commonly used to treat inflammation and the tumor necrosis factor that's found within the body. Okay.
Do you want to bring up the Humira thing and just connect
Sure.
it? We've talked about this so many times in Journal Club already. So I just want to keep bringing it up. This is how Journal Club works. You know, you're not going to get it on the first round, but when you hear the same thing 5 times, you'll be like, oh, that's right. I remember talking about that.
So dexamethasone is a steroid that's used like in the treatment Humira that treats rheumatoid arthritis.
No,
right?
no, not Dex. No, Dex is a different one. So I don't know how often it's used in humans. It is used in humans, but it's actually used in veterinary medicine more. When you're having an inflammatory response to something, dexamethasone is one of the first anti-inflammatory steroids, right? So the difference between an anti-inflammatory that is steroidal, like dexamethasone, and non-steroidal anti-inflammatory drugs like naproxen, ibuprofen, aspirin, meloxicam, celecoxib. Those things are not steroids. They don't bind to steroid receptors. They're not lipids. They are just simple organic molecules. Humira actually interferes. It's an antagonist for TNF alpha, tumor necrosis factor alpha. And I'm on that medication. That's the one that I keep showing you guys. It's $2,500 every time I take it. And I don't pay for it. Insurance does. And that's the thing that causes me to be immunocompromised. And it's also the thing that would increase the likelihood that I would get cancer if I got cancer. If that makes sense. That the cancer would be harder to fight off. Okay, go ahead.
All right. Moving on to the images of the macrophages. So when there is inflammation, macrophages move to the inflammation signal, and the different treatments stop this from happening. So you can see the NFP breaks up the macrophages, and the actin red 555 also changes its structure, and the combination of them also has a greater impact in accordance with fcbd. Probably. You're muted.
Actin red is a stain for actin. Okay.
Oh, okay.
We all know what actin is. It's just a stain. And And 555 means 555 nanometers. So this is an immunofluorescent image. So over the summer, what we'll be doing is something kind of like this, but instead of doing fluorescent, we don't have, if I were to buy a fluorescent microscope, it would cost like at least $25,000. I don't have that lying around. But I'm going to do it a different way, using different other stains and antibodies and stuff. But this thing, actin red, will bind to actin, and it will fluoresce if you use a light that's in this Psi3 red wavelength. So what all you're doing is you're comparing the top row versus the bottom row.
Okay.
And you can see there is a major difference between the two.
Yeah.
Now, before this meet started, Sienna and I were trying to figure this out, and I cannot tell you, I wasn't able to figure this out. To me, I think it should be backwards. If cannabinoids, follow me here, okay? If cannabinoids are anti-inflammatory, then they should inhibit macrophages from wanting to move in different directions. And it's because they want to move in different directions that they form these little filopodia and lamellipodia that go, and they'll actually, these projections will move the cell from place to place. So why would they have more of them? That was where I got stuck. I'm not sure how to interpret that. So sorry, Sienna.
It's okay. I had a hard time understanding it as well. Okay, so this slide just shows all the anti-inflammatory genes within that the CBD is treating, and again, in comparison to drugs that are usually used for treatment and a control. Yeah.
Sam, you missed a veterinary reference.
Oh, I'm sorry. I just got back home. What was the reference?
Just to the use of a steroid, dexamethasone, in animals for anti-inflammatory purposes.
Oh.
You know what you should buy, Sam?
What?
You should get a copy of this. It's really amazing.
Okay.
And
Noted.
the steroids that we're talking about in this paper are used quite often in this book, and every good veterinarian has this. And
okay, I'll get it.
Sorry, Sienna. I think that was trying to make connections here.
Good. And the table is just outlining the size and the size of the macrophages in the last slide, like the images of it, of the control, the fcbd, STD, and just the CBD. And so the conclusions are not written on the slide apparently, but
Is it animated?
maybe. I don't.
I just love that thing, that drawing has made it this many years into Journal Club. I put that on Jordan Amato's Journal Club when she did the very first one, because the paper she covered was so bad. This is such a bad paper, and you guys still use it. It's amazing.
Okay. So the main takeaways are that the strand of the arable strand of cannabis increases interleukin 6 and 8, both of which fight infection. Cannabis is already a medicinal treatment for many afflictions, and COVID-19 may be the next one. And since it's so prevalent right now, it might be the most important one. But and in the paper, it suggested that cannabis may be a superior treatment of lung inflammation than the leading drug.
But not tested in humans, I would think, right?
Yeah, it hasn't been tested in humans yet.
So a bunch of us have had COVID. Did anyone have it really bad? I think we can talk about it freely now, right? I'm not going to get in trouble by being like, you outed my child who had COVID. Like, everybody has it now. Did anyone have it really bad that they had a lot of trouble breathing? Nobody.
I know my dad had it really badly.
Oh, that's right. Your dad did have it bad. I forgot about that. He was in the hospital, right?
Yeah, for a while.
So can you imagine telling your well, then that's appropriate. Can you imagine telling your father, "Hey, I've got some weed. Would you like to feel a little better?" That's nuts to me. Maybe I'm just old. That's crazy. I mean, when I had it 2 weeks ago, 3 weeks ago, whenever it was, I really didn't have. I got a little short of breath every once in a while, but I have an inhaler, so I didn't. Cocaine helped a lot, but other than that, I was fine. I'm kidding. That was a joke for people who don't yet have me for cocaine jokes. Okay. So, is C. sativa a mermaid?
A mermaid?
Well, it says C. sativa.
Oh, Ariel. Yeah.
Yeah. So that you meant arbol, right? I think
Yeah, I
arbol corrected you. Let me be a little picky here, especially for the freshmen. Genus and species names are always italicized. The genus is capitalized, and the species is always in lowercase letters. So that would be cannabis sativa. But isn't sativa's not a species? Sativa's a breed, right? A strain.
Yeah, I think so.
Did you find anything else in the literature that would suggest that they went any further with this in human trials?
No, it actually goes into how it should be there is a need of caution proposing cannabis as a treatment for 19, but it doesn't really go into what that caution is.
I would think so. Again, here, Grandma, have a joint. Father Brian had it really, really bad. I think he had it twice. I think I had it twice. The first time he had it was around the time I had it the first. Well, when Sam tried to kill me. And
Stop spreading rumors.
I'm not spreading it. Everybody knows now you're guilty. The second time, and it felt exactly the same, but with him, the second time was way worse because it was like he says his lungs were on fire. He had so much trouble breathing. I'm actually looking it up. Let's see. I'm gonna say sativa. Somebody raised a hand.
Yeah. So can you just use edibles if their lungs hurt?
Yeah, that's
So
what I think.
Then the liver will break it down.
Yeah, I did. I don't know why I didn't think of that. That's a good point. Like gummies or something. But these data would suggest that it's not that THC is not having an effect, right?
Yeah, I don't think THC is part of it.
Sativa.
So is it just the CBD? They could just take CBD, like the oil.
Would you say oil?
Yeah, there's CBD oil.
Put it under their tongue. That's right, sublingual. So there's a bunch of papers. There's 15 results. So one of them says from Cosdare, possible therapeutic effect of Nigella sativa and its thymoquinone on COVID-19. Then this paper. Oh. So it says cannabis sativa modulates ACE. It's an ACE inhibitor. ACE is a major inflammatory cytokine. I think cytokine again. Yeah, because like what was killing people early on from COVID-19? What was the thing that was killing them? Was it the virus?
Wasn't it the complications that occurred after,
Right. Right. Specifically, what were the complications specifically?
like bronchitis and
Like pneumonia.
Pneumonia. And what was causing the pneumonia? I mean, sure, the virus, but there was an intermediate step, right? Your immune system needed to fight the virus, and so you got this. It freaked out. Your immune system freaked out and released cytokines everywhere, all over the place. And I think I'm pretty sure that I've mentioned this before, but you get this really bad thing called a cytokine storm, where your body overproduces cytokines, which are highly inflammatory, but also extremely painful. And you're in so much duress that you just pretty much expire. Like you gotta have a heart attack and die. And it says that sativa might actually be able to tame those cytokine storms. Is there anything that weed can't do? What a different time. Like we can talk about this now in a way that I could never talk about it when I was your age. Because it was just like, say no to drugs, end of story, that's it. Even as we were starting to get data that it had some potential. Comments? Sienna, other thoughts?
I'm sorry.
Other thoughts?
I don't know. I thought it was like a really interesting like study. I feel like it definitely needs to be like taken further for it to like have any like real implications. But I thought it was pretty cool.
And this is only done in cell culture. Okay. Johan's next week, I believe. Is he in here? Can we spread the word and make sure he knows so he doesn't miss it again like he did last time? Any questions, comments?
so weed is good.
That's a great thing to say. I heard weed's good.
Moral of the story: do drugs.
What'd you say, Tyler?
Moral of the story: do drugs.
Do drugs. Jeez. You know, I post these online, and that's all I need is for parents of young children to be like, I don't want to send my child there. He tells them to do drugs. No, but I'm not going to lie to you guys. There seem to be some chemicals in that plant, the cannabis plant, that do have some beneficial properties. I mean, there are beneficial properties in poppies. Doesn't mean I want you to go do heroin or opium. You know, like yeah. So, I don't know. Don't prescribe it to yourself and be smart about it. There. That's the best I could do. Okay. Sienna, good job. Yay. All right. There isn't anything any further. I'm going to let you guys go. Johan's next week. All right. See you later.
Thank you.
Bye.
Thank you.
Good job.
Bye.